morning. So thank you for this night talk and for being with us at this late time in Korea. And now it's time to go to the second of our speaker with Jean Clairambault, and who has been he's from France, and he's been influential in discussion that I had and helping me to getting introduced to mathematical oncology. So it's a pleasure for me to have him here. So Jean Clairambault is Emeritus Research Director at Inria, and is based at the Laboratoire Jacques-LouisLions of Sorbonne University in Paris, which I think is one of the most important applied mathematics departments, certainly in France, but also in the world. And something peculiar and I think very interesting about him is that he studied both mathematics and medicine. So his career is in math. He does have a real and complete physician's diploma, which makes him see these issues both from the mathematical side and from somebody who knows medicine. He's been the PhD advisor and co-author of Camille Pouchol and the postdoc advisor of Cécile Carrère, whose contribution to AT modeling I mentioned on Monday, and maybe we'll see things that are related today. He has lots of themes of interest, including the atavistic theory of cancer, optimization of cancer treatments, evolutionary approaches to cancer, and drug resistance. And today, he will speak about plasticity in cancer cells and emergence of drug-induced resistance. What consequences for therapeutics? So, Jean, thank you. The floor is yours.
Thank you, Yannick. Can everybody hear me? I hope so. So this is, if everybody can hear me, at least, joint work with people, some of whom you mentioned, mainly Camille Pouchol, and also a former Laboratoire Jacques-LouisLions head Benoît Pertam and present Emmanuel Trella, and others who are either postdocs or PhD students. So I will speak mainly about plasticity. As you will see, cancer is still a puzzle for many people, and I will go certainly beyond what I call intracellular signaling pathways, which is very often the preference of pharmaco-oncologists, and I'll try to go beyond this, admitting that you cannot find usually determinants of cancer in a single cell, because cancer is a disease of multicellular organism, and in this multicellular organism that we are all, cancer may be seen as a localized loss of cohesions between cells and tissues and loss of control of differentiations prior to uncontrolled proliferation. And this sends us to the...
Sorry.
Yes?
Just one thing. Do you have slides or not? Because we don't see slides, so we don't know if you have slides.
Yes, I shared my screen. Can you see my screen?
And so apparently other
people... I was sharing my screen. Is it something that went wrong?
Yeah, no, normally you should have partager l'écran. Okay, sharing your screen on the...
Partager l'écran, so I share the screen. Partager l'écran.
That's somewhat.
Yes, I authorize Zoom to share my screen. And now, well, I could see my... how should I do this?
So, well, when I talk, I just want to then you need probably to see which documents you're sharing. You probably have a choice.
Yes, then I authorized Zoom to share my screen, but it didn't show.
Okay.
Can you see anything?
No,
Optimize
I can't.
Screen sharing.
I see you. Well, there's a backup possibility if you send me your slide. You send me your slide, and I try to share.
I can send you my slides by by mail. Is it possible?
You can try that, and I can try to share my screen and you tell me when I have to switch the screen.
I will do this.
I guess I can do that because I probably have the right to do that.
I hope so.
Or you can close Zoom and restart. You can also try that.
I'll do both if you want. Okay. To Yannick.
Yeah, that's what Clairambault says. Clairambault says, try stopping the shares and sharing the whole desktop.
So should I stop everything?
Yeah, you can. Yeah, you can try stopping the share, he says, and sharing the whole desktop. I'm not sure exactly how you do that, but try to do that. On my side, I will try to get your slide and.
Well, I'll send you my slide first.
And are you using multiple devices right now?
No, he's using. I don't think so. No.
I was first trying to share my screen, but what happened is when I click on share screening, then authorize Zoom to share your screen, and I have to open system preferences, which I cannot see. Let's see.
So we're still learning to do this online workshops. I guess I should have asked Jean his slides before.
Okay.
Indeed, probably in system preferences, you probably need to give
so
some right to Zoom.
Should I quit and reopen?
Yeah, try that. You can try that. So apologies for that. And by the way, if the next speaker is worried that some issue could happen, so Eduardo, if you want to send me your slides, that way I have them and I can always try to share them myself or to give them to Kevin from mydepartment.org to try to settle any possible issue. Don't hesitate to do that, and that's true also for the people who are speaking this afternoon. If they're worried about a similar issue occurring. So on my side, I will see if I receive anything from Jean, but I guess we'll manage to deal with that situation. So let me take this opportunity of a few minutes to say that there is a discussion session later today. So feel free, if you have topics that you would like to share with us, to send us emails saying, can we talk about that, etc. But of course, everybody will also be able to intervene and we also plan. Oh yeah, that seems to be better. Oh, what's happening?
It's dropped again.
Yeah, I don't know. Well, I was hopeful, and we think that even though it's not over, and we're having some technical issues, there was a lot of people who attended this meeting, and so I think this gives us encouragement to try to have some other similar meetings. So we have already a few people who express interest in hosting such physical such meetings. Maybe next year or in 2 years physically. Maybe there will be also other virtual meetings. But we hope to keep building a community.
Jacques-LouisLions and Trigos recently, and I will try to by the end of this talk present. Ideas about what is this coherent or cohesion between cells and tissue made of in a multicellular organism, and how is it disrupted in cancer. So cell plasticity is the main thing that will explain, from my point of view, the drug tolerance or drug resistance in as much as it is drug-induced. And for this, we'll have to resort to mechanisms of the great evolution, that is slow genetic mechanisms, but from the point of view of cancer, which may be seen in the atavistic theory of cancer as a fast reverse evolution on smaller time windows at the scale of human life, it will be a way, a process, a reverse evolution process that will be able to explain much of plasticity. And for this, intratumor heterogeneity must be observed from the point of view of cancer cell populations in which plasticity, that is propension of epigenetic nature to reverse or to a dedifferentiated status, will make the cancer cell population able to first develop phenotype switches such as EMT, but also to reversibly resist a dropped drug insult by resorting to sharp stress response mechanisms. And what is interesting from my mathematician's point of view is that such reversible plasticity is captured by mathematical models based on PDEs that incorporate between cell population heterogeneity by continuous phenotypic variables. And you will see that one of them may be considered as a continuous drug tolerance phenotype. And moreover, you can use optimal control methods to design therapeutic strategies. Well, I will skip very briefly on this, just to say that we should exclude the Luria-Delbruck mechanism because bacteria are not cancer cells, which are much smarter, that is endowed with a rich genome, and that makes them able to very expanded plasticity. And this paper by Sharma in Cell 2010 from Jeff Stattelman's laboratory showed us that when you submit a population, when you expose a population of very aggressive cancer cells, NSCLC cancer, lung cancer, then you can see at the beginning that most of these cells survive, die, but a very small percentage, a tiny percentage of them survive, which people using terms from bacteriology name drug-tolerant persisters. And after some time, second phenotype emerges, and you can see drug-tolerant expanded persisters that is within the same bath of the drug, a very high dose of drug. Here it's gefitinib, but it may be others used in these publications. And then you can see that these cell populations become completely tolerant to these drugs, which is completely reversible if you withdraw the drug from the bath. And this takes some time, but the reversibility seems to be linked to an epigenetic enzyme that, when it is blocked, blocked the emergence of drug-tolerant persisters. So, is it genetic mutations, as is the most admitted mechanism, or is it phenotype switching? In what I have just presented, clearly it's phenotype switching because it is completely reversible. But it cannot be excluded that after some time, drug tolerance leads to mutations that make the cell population drug resistant, if we use precise vocabulary. And in fact, there are biological observations that support this point of view. But from now on, I will use drug tolerance in the general sense of drug resistance equally. So what about the atavistic theory of cancer? First of all, you know this motto by Theodosius Dobzhansky, and it induces us to see that neoplasm, which is the usual word for cancer, in fact should be called more archaeoplasm. This is a word from Mark Vincent. And you can see that from the point of view of the great evolution, mammalian capabilities in our cancers go down to multicellular metazoan capabilities that are certainly developed, but not enough developed, of course, as the very evolved multicellular organisms we are all. And this is documented, as I said, in different papers based on biological observations, and in particular by these Domazet, Locho, and Tautz, and Trigos also, which underscore the fact that genes that have appeared in the development of multicellularity are precisely those that are altered in cancer. So a question might be, what genes, knowing that the evolution towards multicellularity begins by colonies endowed with proliferation mechanisms, later came apoptosis, and later, much later, came cell differentiation and division of work, and much later even came epigenetic control. And this you can see in a reverse mutation order in acute myeloid leukemia samples, fresh blood samples in patients. That is, first, the mutated genes are the epigenetic control, then the cell differentiation genes, and finally proliferation. So it's a decomposition, a deconstruction of the whole building of multicellularity. So these also must be seen from the point of view of different metaphoric representations. Let me underscore the fact that the celebrated Waddington epigenetic landscape is not at the level of the big genetic evolution, it's at the level within a given organism of cell differentiation. And it has been redocumented by systems biology models, by Sui Huang in particular. And here at the top of this scenery, you can see the totipotent cell that finally gets to completely differentiated cells at the end in these attractors of mature cell types. I will propose another phenotype, metaphor, which to my meaning is new, as a construction of a multicellular organism as a fiber bundle with base, the body plan, fibers, the cell differentiation trees and twigs, and at the rim, that is here in this metaphoric representation, the terminally differentiated cells. So what means the fact that there is cohesion in this multicellular organism is represented by what I call the cohesion watch, which is certainly acellular and not identified so far. But these three elements, body plan, differentiation trees, and cohesion watch, together make what is maybe called a Borromean knot. So enough about metaphors now. Let's speak a bit about mathematics. So structured equation models for heterogeneous populations are very simple in this presentation. You have a population constituted of population density here of cells at time t and a bearing trait that is phenotype X, and you have the evolution in numbers of all individual constituting the population if you integrate over the phenotype, over the trait, which allows you to follow the evolution in numbers. You also can follow the asymptotics and the distribution, the evolution with time of the distribution of the trait by following this probability density function. So tumor growth may be followed from the first point of view, and the distribution of trait and its asymptotics from the second point of view. So what is the asymptotic behavior of the total population? What are the fates of the phenotypes in the population? To document this, the main ingredient is a non-local Lotka-Volterra integral differential model, to which one can add later second order terms of diffusion or integrals with the kernel. But the main ingredient here is the competition term which is non-local, which is a logistic term here. This means competition for nutrients or maybe space, if your cells are in contact, of one cell with all the other cells. This is what is represented here. This is a basic principle of the non-local Lotka-Volterra integral differential terms. And you can see that with very simple hypothesis, the total population always converges to a plateau, and the phenotypes evolves following either no pressure by drugs or anything, or following a trait. Here there's no pressure, and you can see that following in time the population, it converges, it concentrates in trait to here x equals 0. So this is documented in the paper by Camille Pouchol of 2018. Now, what you can do is, more interestingly, about cancer, considering two different populations, one cancer and one healthy cells. Because when you do therapeutic representations, you always act on both healthy cell population and cancer cell populations. So we want to take into account healthy cell, that is unwanted toxicity on healthy cells. And also we want to take into account plasticity, that is adaptive dynamics within cancer cell, the cancer cell populations. And for this, we have to take into account the total population. Here it's not raw, it's linear combinations of the two populations, row H for healthy cells and row C for cancer cells, with a well-chosen combination terms. And then you can add representation of the drugs. Here, supposed to be constant. We will see later how they can be made variable in time. One is a cytotoxic drug acting on an added def term, here U1, cytotoxic. And here we just have another cytostatic drug used in combinations. For instance, it may be a drug that acts on a receptor, on a proliferation receptor acting on combating a growth factor. This is a targeted therapy, if you want. And this is chemotherapy. So you can now act with these combinations to optimize this. And this is just a pedestrian optimization scheme where you can see that cancer cells here get extinct with the chosen, well-chosen coefficients, of course, whereas healthy cells do not get extinct and survive. So this is a representation of what can be done. And here now comes the optimal control problem. This is exactly what I presented in the previous slides, but U1 and U2 now are dependent on time, and you have to solve the optimal control problems. That is, in a fixed horizon T, minimize this cancer cell population, total cancer cell population at time capital T, with U1 and U2 being two L2 functions depending on time of drug administration. And since it's optimal control, it's natural to add the constraints. And these constraints are mainly addressing healthy cells. So you don't want to be too damaging to healthy cells, and you have the objective functions, which is here, this CT of U1, U2. Now, what must be absolutely avoided? How to be deleterious, that is pessimal solution, by using constant doses, high doses and constant doses of drugs. What you can observe in these simulations is when you do this, constant doses, high doses of U2 mainly, that is a cytotoxic population, is that you concentrate the phenotype of drug resistance or drug tolerance on a very, very resistant population phenotype here. And you observe that after some time, the population of cancer cells having been firstly decreased, increases without limits. And it's also catastrophic for the healthy cell populations because they also decrease. So this is what is a pessimal solution. But the optimal control solution consists in a theorem that I will not detail now, but I will rather present to you the optimal control solution illustrated by optimization algorithms AMPL and IPOpt, and here simulated with well-chosen functions and coefficients, where you can see that here the phenotypes that are represented here evolve first in the healthy cell, in the cancer cell populations from here, this, it should be black and then become red. But the main difference here with the healthy cell population is the black curve here becomes red and stays as a sensitive phenotype and not at a drug-tolerant phenotype. But here it's flat because what you can see is that the row C population converges to 0. And most important is the fact that the solutions are here, U1 and U2. U1 is a cytotoxic drug solution function, and you should give approximately nothing for most of the time. That is here 2/3 of the horizon T, so as to let the cell population become of a very sensitive phenotype before you administer the maximum tolerated dose for a very short amount of time. And for the cytostatic, it's not so important because you have here the possibility to be constantly at the maximum tolerated dose for the cytostatic. But the most important is that the cytotoxic, the chemotherapy, should be used for a very short amount of time and then set at some midpoint, which will yield a healthy cell population set over a given threshold. So this is what comes out of the optimal control solution for a given horizon. Now, almost periodic therapeutic strategies give us the non-satisfactory solution here that you decrease but re-increase again. Whereas here, what I try to, yes, when you try to do a second course of chemotherapy, what you can see is that, and this should be again more explored, it has been just approached in our first simulations from our models, is that it seems that using the same strategy, but with saturation of the constraints, rho h equal theta h rho h of 0, that is giving the maximum possibility of damaging healthy cells, then it seems that you can go to maybe towards extinction. And the hope is that by this sort of adaptive therapy, you can gain maybe close, you can go maybe close to extinction. So this is what comes out of our, sorry, what comes, therapeutics? Oh, yes. So, yes, how much time do I have, Yannick?
Well, you can still use 5 minutes and then maybe we won't have so many questions, but that's okay.
Yes, but yes, well, I would just wanted to know that, to mention that what is very important is that in this stress response, we have just seen one phenotype, that is drug tolerance. But you can know that I have tried to make you feel that the most important thing is plasticity. And plasticity may be expressed by different phenotypes. And this may result in bet hedging as a tumor strategy. And this is a field that we are exploring now and trying to find not only drug tolerance, but to explore different ecological phenotypes that are usually found in ecology theory, fecundity, viability, to which I have added plasticity. And plasticity has some diffusion, ability to diffuse in phenotypes. And here, what we are trying to do is trying to find something like a possibility of divergence of phenotype that could explain why certain cells in cancer cells that are submitted to high stress should choose one phenotype and some others another phenotype, which could also send back to the origin of multicellularity, which may be seen as a divergence of phenotype. So, well, for the immune checkpoint inhibitor immune therapies, it's the same. They are also cell-killing therapies, and we should be confronted with exactly the same problems with optimal control. Now, I have briefly mentioned this idea that multicellular organisms are constituted by a base, which is the body plan, fibers that are the differentiation trees and twigs, and also what I hypothesize only, I have no documentation in a molecular way of what it could be, a cohesion watch. And so if we could identify such a acellular cohesion watch made of intercellular signals that make cell populations cohesion together in the multicellular organism, then we might work without necessarily killing cells, but obtaining cohesion in the multicellular organisms that bear cancer, which is disorganization of multicellularity. So to find new therapeutic tracks, we should certainly make use of cell-killing therapies, but using optimal control models in heterogeneous cell populations. And of course, immunotherapies are no exceptions, and we still have much to understand in what makes them work and what makes them fail. William Coley is an example, but of both. So we should be able to understand better what is a multicellular organism and what is disrupted in its coherence when cancer comes. And it's not only speculations. I think that if we have this in mind, then we could find other ways of having cancer treatment strategies. So these are a few references. And if you want to also, an easy reading conference paper that was presented last month or in October, sorry, in San Diego. So thank you for your attention, and I can go to answering questions if there are.
So thank you, and there are indeed some questions. So there's a question by Jessica Cunningham, who says, absolutely fascinating talk. Have you considered other objectives? For instance, minimizing specifically the resistant phenotype, or a favorite, minimizing tumor variance over time?
The resistant phenotype here is X in this simplified vision, which is the drug tolerance or drug resistance, and all cells are endowed with this phenotype. And here we see the population that will be adapting itself to this evolution of the phenotype under the pressure of the drug. If there is no drug, then there is no pressure, and you will see the population evolve toward total sensitivity, that is X equals 0. When drugs come, and we have favor here, the cytotoxic drugs, then the population, as I showed you, for instance, in this pessimizing solution, will go to enhanced drug tolerance here close to x equal 1, which is maximal drug tolerance. X equals 0 is maximal sensitivity. So this is precisely where the drug tolerance phenotype is represented. And as I said later, if we want to have finer phenotypes that is related to fecundity and plasticity and viability, then we'll have to represent things in not only in one dimension, but in many dimensions. But it was easier, of course, to use this drug tolerance phenotype, which structures the population completely in its heterogeneity, in what we have represented here. So is this some answer to Jessica, or should I?
Yes, yes, she said in the chat that she misunderstood before, and this is great. Maybe there are more questions, but maybe I'll just ask, I'll just take one more question in the interest of time. So it's a question by Fred Adler, who says, this is a beautiful modeling approach. Does this thinking lead to different predictions of organs most likely to get cancer than the standard approach of counting numbers of cell divisions?
Well, what I understand in Fred Adler's question is about the constitution of multicellular organisms and where it is concerned by cancer. In my representation, my metaphoric personal representation, cancer occurs somewhere in these fibers, which represent trees and twigs of differentiation in the multicellular organisms, where some control is deficient. And where this control of differentiation is deficient, then cells are blocked in this control and they can start proliferating. And of course, they're blocked in their differentiation process. So they are plastic. They are very plastic because their terminal differentiation is at the rim. Or if you want, also in the Waddington representation at the end of these mature cell types. And so you have representation of at very specific, starting at least, because after that you have metastasis, but at very specific functional and anatomic points in this representation of the multicellular organisms from which cancer is able to start. It can be, by the way, at stem cells, cancer stem cells, but not necessarily. It can be anywhere in the cell differentiation process. So this is both an anatomic representation and let's say differentiation representation of the cancer process. I'm sorry to say that it's still metaphoric, but maybe dealing with geometrical approaches, we can make this more sound. Is this an answer to Fred Adler, or do you want, Fred, something more precise?
Yeah, well, I mean, if we want something more precise, I think we'll have to take it offline because we'll have to proceed to Eduardo's talk. So thank you, Jean, again. And so there were